It's my great pleasure to introduce Dr. Chris Hsiung, who is an assistant professor with a joint appointment in the Institute for Human Genetics, as well as the Department of Surgery at UCSF. Chris earned his bachelor degree from UC Berkeley before he joins the MD PhD program at the University of Pennsylvania. At UPenn, he was co-mentored by Gerd Blobel and Arjun Raj, focusing on how gene regulatory information is preserved or altered through mitosis in mammalian cells. Following his graduate training, he completed his residency at Stanford University. Meanwhile, he was pursuing his postdoc research at UCSF as well as Arc Institute. His postdoc work with Luke Gilbert mainly focused on how to develop novel CRISPR-based tools for high-order combinatorial perturbation, as well as deploying CRISPR screens, especially the CRISPR screen platforms to discover genetic dependencies in immune recognition of cancer, as well as alteration in cancer cell differentiation state. Chris has received numerous honors, including the Prostate Cancer Foundation Young Investigator Award and the NIH KO1 Mentored Research Scientist Development Award. In addition to his research accomplishments, he has also been a very committed mentor to many PhD students as well as postbac research scientists throughout his whole training. So please join me in welcoming Chris as he presents today's seminar on Cas12 for high-order chromatin perturbation. Welcome, Chris.
Thank you, Sky, so much for that really nice introduction. Hello, everybody. I just started my lab in November in the Institute for Human Genetics at UCSF. And this is my new lab members. And I would say, broadly speaking, our interests are in pondering the flow of genetic and epigenetic information through biological systems and trying to tinker with that flow potentially for therapeutic purposes and as well as basic understanding. So, because Sky already gave a wonderful introduction, I did my MD PhD with Gerd Blobel and Arjun Raj, focusing on genome regulation during mitosis. And then my postdoctoral work was with Luke Gilbert, which is actually most of the talk will be about the work that I did there and is now the foundation of my new lab. So I'll describe to you our interest in functional genomics, which I will define as a specific implementation that you all know as basically CRISPR screens, right? So this is an approach to genetics where we are able to introduce many perturbations in the form of guide RNAs in a pool and then read out the effects of those perturbations at the phenotypic level, often using sequencing. So I think the key to the scalability and success of these methods is that we're encoding both the perturbation and the phenotype as nucleic acids. Uh, and and so this approach has already been quite successful. Of course, you know about it as targeting genome-wide coding genes, but the first CRISPR therapeutic, actually, the development of that therapeutic was in part contributed to by using this kind of approach in non-coding regions of the genome, and also resulting in the first CRISPR therapeutic is also the first enhancer therapeutic, and that treats hemoglobinopathies. And today, what I want to focus on is how can we increase the number of perturbations we introduce into each cell, and especially going to 3 or more perturbations. So why might we want to do that? So here's an outline of my talk. I'll talk a little bit about how I think about combinatorial genetics, and then introduce a new platform that we developed to address some of those challenges. And then lastly, go over a conceptual problem. How do we efficiently search for through numerous combinations of genetic perturbations? So I think one example that I really love about that illustrates the necessity of exploring combinatorial genetics is from the discovery of the IPS reprogramming factors from Doctors Takahashi and Yamanaka back in 2006. So This is where they put uh, candidate transcription factors into uh, uh, fibroblasts, and they saw that they were able to reprogram them into iPS cells. But interestingly, they were not able to do this with two factors. They really needed to go to three or four factors in order to achieve this. So this is a really nice example of an emergent property that arises from combinatorial genetic perturbations. And that's in the space of the coding genome. What this kind of uh, combinatorial interactions among genetic elements is, of course, also very important in the non-coding elements of the genome. And as you all probably know, many of the genes in our genome can be regulated by sometimes more than one potential non-coding regulatory elements, such as enhancers, shown here just for one example at one locus around the MYC gene. And on average, there are about four predicted potential enhancers per gene in the human genome. And the question that we have is, how do we discover and engineer these at scale? Right? It's known that often when you perturb one of such candidate enhancers, you don't necessarily get the phenotype, the impact on gene expression, and that there is some amount of functional compensation or cooperativity among enhancers. So one major challenge is to be able to discover them and to engineer them uh, at scale and try to understand their combinatorial logic. So that's in the next part of the talk, I will discuss a platform that we developed to where we've tried to address that a particular question in particular. So this is published in 2024, and our constructs are on Addgene, so feel free to look there or also reach out if you have any questions about this. So our goal is to develop a platform where we can encode many perturbations that are linked on a physical construct and conduct these high throughput pooled CRISPR screens. And in order to achieve this, we wanted to use CRISPR-I as a tool, which you've already got an introduction to in the previous talk by Danica, where we fuse Cas proteins to the crab domain, which we're borrowing the ability of such domains to recruit the endogenous repressive machinery of the cell to induce the formation of heterochromatin at the targeted locus. And this we chose CRISPR-I because this avoids excessive genotoxicity that might occur if you otherwise chose to use a Cas nuclease, for example, to target multiple sites in the genome. And another reason is that we wanted a tool that is effective at not only targeting genes, but also non-coding elements such as enhancers. And this is thought, previous work have shown that CRISPR-I is generally a bit more effective than nuclease-based approaches, probably because the crab domain, by recruiting the repressive machinery to induce heterochromatin, affects a larger area around the chromatin, whereas cutting tends to be, uh, you need the PAM to be sort of positioned uh, immediately next to maybe transcription factor binding sites that are important for enhancer function. So we chose CRISPR-I to pursue, and the challenge with Cas9 based approaches has been that it's difficult to have more than two perturbations per cell in these high throughput screens, and I'll describe a little bit why. So Cas9 guide RNAs are fairly large. They are not quite as suited for multiplexing because each guide needs to be, in general, needs to be encoded under a separate promoter, and the scaffold region of the guide is also fairly large. So in a hypothetical six-plex guide array, you would need around 2 kb to encode a six-plex guide array. That introduces many challenges for cloning, as well as when we package these into lentiviral particles, there's recombination that can occur in that process due to the homology that's introduced in these large arrays. So in order to overcome this, we chose to go with a different enzyme called Cas12a. This enzyme has been around since around 2016, and immediately it was articulated to be uh, people have thought that it is good for multiplexing. And the reason is that Cas12a has its own RNase domain to cleave the pre-CRISPR RNA and allow the maturation of the pre-CRISPR RNA into mature guide RNAs. And because of this, one can encode Cas12a guides in a very compact array where the each spacer is only 23 nucleotides, and that's preceded by only a 19 nucleotide direct repeat variant, compared to like the Cas9 scaffold is more than 90 bases. And also, we can encode this whole thing under a single promoter because of the RNA's activity of Cas12a can maturate each of these guides on its own. So for a sixplex guide array, this comes out to be around 252 bases compared to more than 2 kb for Cas9. Um, when we started this project, there were no CRISPR-I pooled screening platforms, however. So we set out to build one. And at the time, Cas12a had already been optimized a bit for nuclease screens. And here I'm using the nomenclature from previous work by others, where this EN stands for enhanced. It includes three mutations that people have already introduced to enhance affinity of the Cas12 protein and to improve cutting. And AS stands for the bacterial species that it came from. And so that has already worked for high throughput pooled nuclease-based screens. And we at the time we simply wanted to inactivate the DNA's domain of this protein, and basically this represents the state-of-the-art optimizations of the field at the time. And we fused this to the crab domain. So we performed an experiment where we targeted 500 or so essential genes in K562 cells, and we tiled the TSSs of these genes with single Cas12a guides and compared to negative controls. And here we are using essential genes because we're using cell fitness as a proxy for the CRISPR-I activity of these guides. And on the y-axis here is the cell fitness as a cell fitness score as a proxy of CRISPR-I activity. So the lower the y-axis, the stronger the CRISPR-I activity. And on the x-axis, I'm showing you a metagene profile of um, the position of the sites that targeted by these guides relative to the TSS, averaging across all the guides targeting all the genes in this library. And on the right panel, I'm showing you a control of using dCas9 crab. And this shows you a bimodal pattern where you get the strongest activity proximal to the TSS with a dip in activity around 150 bases or so after the TSS. And this has been previously shown to be correlated with probably occlusion by a well-positioned nucleosome in that region that inhibits Cas9 binding. When we did the same assay with this DNA's inactivated version of EN-Cas12a crab, we get a similar pattern, but notably this is really, really very weak and unacceptable using this assay. And this was somewhat not a surprise. It was kind of an open secret in the field. Other people have also observed weak CRISPR-I using DNA's dead versions of Cas12a, especially for functional genomics applications where we are delivering the guide as a single copy integration um, using lentiviral delivery. So it's a very stringent and dose-limited application in order for things to work for high throughput pooled screens. So we wondered why, because EN-Cas12a, the nuclease itself, already works quite well for high throughput pooled screens, and we set out to try to solve this problem. So there was about a year of work where things didn't really work. We tried to introduce a variety of mutations into this protein, but here I will tell you, um, we had to dig into the biophysics literature a bit to try to solve this problem. So in previous work done in vitro, it was shown that Cas12a basically can complex with the guide RNA and through a process of R-loop formation can bind to DNA. And in a DNA's dead mutant, so the DNA's active site is in the RUV-C active site. When we inactivate the DNAs, these two configurations are the only states that are available for the Cas12a to bind, and the R loop can reverse itself because the non-targeting strand of DNA can actually sort of zipper back with to bind the target DNA strand to compete with the guide RNA and reverse the R loop. Okay, and in a wild type enzyme, the enzyme actually makes a first cut in the non-targeting strand and then makes a second cut in the targeting strand to make a double-stranded break. It turns out that through reading this biophysics literature, it was shown that once the Cas12a enzyme makes this first cut, it's much more stable on DNA. And so we thought that maybe if we can engineer that um, to trap the enzyme in this state, we might be able to allow the protein to bind longer and allow transcriptional repression to occur more effectively. So we introduced a mutation that was only previously characterized in vitro, and this mutation basically slows the cutting of the target strand significantly. And basically, our goal is to trap the enzyme in this nicked state in order for there to be enough time for repression to occur. So we incorporated this mutation into the prior state of the art by replacing the DNA's dead mutation with this R1226A mutation that basically turns the enzyme into largely a nickase. And lo and behold, we see that this mutation rescues the activity of Cas12a in this high throughput CRISPR-I assay. And next, we wanted to show, so that was the first time anyone has been able to perform a pooled CRISPR-I screen with Cas12a using a single guides per cell. But then really the key is to test whether this new enzyme, which we now refer to as Cas12a, can can perform multiplexing. So to test this, I cloned a number of guides targeting a cell surface marker CD81, and the guide is arranged in different configurations going from 1-plex to 6-plex, and I'm encoding the guide targeting CD81 at different positions as shown here on the x-axis. And on the y-axis, I'm showing you the percent expression knockdown of this gene CD81 that we're targeting and comparing each of these different proteins. So the DNA's dead version is the prior state of the art, and then we're using multi-Cas12a, which is the enzyme that incorporates the R1226A mutation and is a nickase biased enzyme, and versus multi-Cas12a alone without the crab domain as a negative control. So here I'm showing you that in this particular case, this guide, you do get some activity with the DNA's dead version of Cas12a, but as we increase the amount of multiplexing that we do, we start to lose activity completely with this DNA's dead Cas12a. Whereas with our multi-Cas12a crab fusion, we retain significant activity, CRISPR activity, even going up to 6-plex guide arrays. And we compared this to without the crab domain, we get almost no activity, so indicating that this is occurring through transcriptional repression mediated by the crab domain. So this is a relief. We were really happy to see this, and then we wanted to ask whether we can, what is the upper limit of multiplexing, right? So to test this, we encoded the guides in a variety of 6-plex, 8-plex, and up to 10-plex constructs where there are a number of guides targeting different cell surface markers in the cell. And on the x-axis, I'm showing you each of these uh, array configurations going from 6, 8 to 10-plex. And on the y-axis, I'm showing you the percent expression knockdown of these individual cell surface marker genes that we are targeting. So as you can see here, for most of these genes, we are actually able to get uh, CRISPR-I knockdown going from 6, 8 to even 10-plex uh, targeting. We do see some compromise in activity as we increase the amount of multiplexing, especially for this guide, which we know from more studies that it's a little bit intrinsically weaker as a guide on its own. And we think that as we increase the multiplexing, we are reducing the amount of protein in the cell that's available to bind each individual guide in the array. So this is something that I think we can expect on based on those biophysical principles. And but however, I think this is really quite promising in that at least for the 6-plex, we certainly have all the guides working quite well, and we still have some activity even at 8-plex. So we then wanted to evaluate how active is each position in this kind of array. So we took the guides that were empirically active in our first pooled screen targeting essential genes in K562 cells, and then we put each of those active guides in different positions of 6-plex arrays. And for the remaining of the 6-plex positions in the 6-plex arrays, we fill them with negative controls that are targeting intergenic regions of the genome that are thought to be inactive. So this was the first time we were able to try doing a 6-plex high throughput pooled CRISPR-I screen. And what we found was that most of the positions retain most of the guides that were active as single guides, we do retain activity when we embed them um, in these 6-plex arrays. So I'm showing you the percent recall of these known active guides in this 6-plex pooled screen. And there is certainly some differences in position, and this is also confounded by the fact that at each of these positions, we've engineered a slightly different variant for the direct repeat so that we can reduce the amount of recombination that might occur when we clone and introduce these constructs as lentiviral constructs. So I think this is a glass half full, half empty kind of situation. We think we're very happy that we were able to get the 6-plex pooled screen working, but there is some compromise in activity, and it kind of depends on which position of the guide it's in. Okay, so next we wanted to, and also because we've engineered this enzyme to have a nicking property, we wanted to show that indels are not accounting for the expression knockdown. So to do that, we measure the indel formation in this particular case, two guides that are spaced around 100 bases apart. And we show that compared to a fully active nuclease, this down here is our multi-cassette construct. We have very little indel formation, although there's still a tiny little bit. And we calculated based on this frequency of indels, the percent knockdown that we would expect, even if these small indels were complete genetic nulls, the most amount of knockdown we would get for this gene would be 1.9% compared to the observed knockdown is actually 90%, indicating that the most of the knockdown is coming from transcriptional repression through the crab domain. Okay, so here I will describe our attempt to use this platform to understand the combinatorial function of enhancers, or at least show that this platform can be used for studies of this nature. So here we focused on the MYC locus as a model locus where previous studies have described a number of known enhancers in this locus. And here I'm showing you just 3 of them. And in the field of gene regulation, people have really pondered what kinds of combinatorial what kind of function do enhancers function in combination quantitatively. And some studies have shown that enhancers can be additive, sub-additive, or super-additive. And in this particular locus, so I think various observations have been made as well with going up to pairwise perturbations. And here we wanted to go up to three plex combinations of enhancers. So to do so, we designed guides against these three enhancers as well. We also tested the promoter as well, and we assembled these guides into using these six-plex arrays into many combinations and permutations that target all of these enhancers and their lower order combinations. And here, because the MYC locus, the MYC is an essential gene in K562 cells, and previous work from Jesse Ingrid's lab has shown that the self-fitness is proportional to the MYC expression in this case. And so we're using self-fitness as a readout of MYC expression knockdown on the y-axis here in our pooled screen. And on the x-axis, I'm showing you the enhancers that are targeted in each of these uh, combinations. So we have either guides, constructs that are targeting just enhancer 1, 2, or 3, or paired perturbations, or up to three-plex perturbations. And each dot here is an individual construct in the library. What you can see here is that we definitely see a cumulative effect of targeting more enhancers, giving stronger CRISPR-I knockdown read out as self-fitness in this assay. And if we stare at this plot a bit longer, you'll find that if you try to add up the individual effects of the individual enhancers, the pairwise ones are a bit closer to additive, maybe a little bit more than additive, but once you go to three-plex targeting, you do get more than additive compared to what might have been expected from additive of this red line indicating the additive of the individual perturbations. Okay, so this suggests that there may be some super additivity once you, in this particular locus with these particular enhancers, going from above to paired perturbations of enhancers. We actually didn't make very strong statements about this in our paper, as our paper was mostly focused on the technology development aspects, but I think we think this illustrates at least one example where this tool can be used to study such questions in enhancer biology more broadly. Okay, so as a summary of our platform, we engineered the Cas12a enzyme using the nicking-induced stabilization, which is actually a novel way of engineering Cas proteins to make them better for transcriptional perturbations. And it works for pooled CRISPR screens, including up to six-plex guide arrays. I haven't shown you that but we've tested in multiple cell types where it works. And it works for combinatorial targeting of enhancers, and in some work that I don't have time to show here, we've used it to discover new enhancers as well. So finally, I will give a quick summary of what I think is a conceptual problem, which is how do we efficiently search large numbers of genetic combinations? So, once we go to up to three-plex, the combinations just explode, right? And or above. So you might ask, like, Chris, even if you can target 6 or 8 or 10, how can you actually effectively explore this huge number of combinations? So here I would like to point out that I think an important concept is that when we test one of these combinations as an array of guides, if you design a screen such that the guides don't cancel each other out in their effect for a particular phenotype, then testing a single one of these n-plex combinations is potentially indirectly also testing its lower order combinations. And for an n-plex guide array, you're testing up to potentially 2 to the n total combinations. So thinking about this concept a bit more, how can we realize the benefit of this? So I was inspired by this concept of group testing, which is a very simple concept that was originally proposed in the World War II era, where this man, Robert Dorfman, was trying to think about how to effectively test many military recruits for syphilis. And now he proposed that you can pool the individuals into groups and test them, and if they're negative, you can just ignore that, and then if they're positive, you can then test that group exhaustively, individually, and in a secondary test to see which who is positive, right? So this is, of course, now widely known as pooled testing in, for example, in COVID testing. And so we can think about this as an analogy. We can treat each of these guide arrays as a group. And if an array tests negative for our phenotype of interest, then we can ignore that and not have to exhaustively test all of its lower order combinations. And if it's positive, we can then do a secondary test to figure out what combinations are positive. So I think what I'm proposing is that we can split these functional genomic screens into two steps where we test higher order combinations first, and then each array is testing 2 to the n uh, combinations indirectly, and taking those hits, we can do a secondary screen to then enumerate the lower order combinations if that's of interest. So this avoids the costliness of exhaustive testing. So to summarize, I think our platform, we've implemented this in some of our enhancer discovery efforts. But in the interest of time, I will just summarize here. Um, I think that higher order multiplexing is an efficient way to explore lower order combinations, and potentially each increase in multiplexing is gives you an exponential increase in the amount of search space that you can cover. And in order for this to work, you need that each guide needs to perform well once it's embedded in the context of an array, that there's no dilution to the signal or interference. And that's really what we've achieved with our protein engineering here, is to make our CRISPR tools more robust to those effects. So uh, thank you for that. So I want to acknowledge the folks who have contributed to this work, including Luke Gilbert, my postdoctoral mentor, and also others in the lab who have contributed to this work. And these are our past and current uh, partners who funded us. So thank you very much, and I'll take any questions.
Thanks, Chris, for the presentation. If you have any questions, please type it in the Q&A box, and so we can read it to Chris for him to answer. So I have a question. For a lot of examples you showed, for example, MYC, these are constitutively expressed genes. But how about like activity-dependent gene, like FOS or some of the immune response genes that are only expressed upon stimulation?
Yeah, I think that's a great question. We haven't tested those currently, but I think that's definitely of interest. I think the genome has been explored largely in most of the time in homeostasis or development, like characterizing enhancer activity. But I think there's maybe more, potentially more elements that might be more responsive to certain environmental cues. And I think this kind of approach, such as like during tissue injury or damage or any of those for example, inflammatory processes. So I think this tool can maybe help us discover those elements as well if you have the right model to put the biological system in those settings.
Yeah, I guess I haven't followed these literatures for too long. Is there any method to efficiently deliver these Cas12 proteins into the primary cells? Because with cancer cell, of course, you can do lentiviral infection, but for example, with neurons, we really can't do high titer lentiviral infection. So is there any peptide delivery method, or is there any knocking mice that people can utilize for primary cells work?
Yeah, so people have there are people the Cas12a nuclease has been put in. There are papers and preprints about transgenic mice that are expressing the Cas12a nuclease. We haven't developed the CRISPR-I version of that yet, but I think that would definitely be an important future direction. Yeah.
So you also got several questions in the Q&A box. The first question is, it seems like the upper limit for multiplexing is also target dependent. Is that true? I think that's a question.
So yeah, so I would say there's a lot that we need to explore to predict guide activity better. So there's two layers to this. The first part is that individual guide activity is actually somewhat difficult to predict still for Cas12a, to some extent also Cas9. Uh, and then once you assemble them into arrays, they can the potentially the pre-CRISPR RNA, there may be RNA folding issues that could alter the activity of the guides depending on the exact configuration they're in. And we detail all of this in our paper. So I think this is really an area that needs to be explored and is really ripe for, methods like machine learning, where if we have large data sets on array guide activity to predict um, the activity of guides. Yeah.
A related question to that is for those guide RNA which lose efficiency in the multiplexing, did you know if it is a limitation of the Cas12 enzymes or it might be the pre-guide RNA, it's not properly processed?
We have not directly measured the processing of the higher order arrays currently. There could also be, once you get to longer arrays, the what we're using right now is the U6 promoter. It could be that it's not making the full length transcript as effectively. And others have proposed like Pol2 promoters as well, but in my hands, the U6 has worked so far. And so there are, so the short answer is we don't know quite yet. We do think that enzyme we have improved significantly, but there could be room to improve further. We've shown that it's still, dose dependent, how much protein or guide, either one you put into the cell will matter. And this is all detailed in our paper as well. Yeah.
Thank you. I think because of the time limit, we are going to end the session for today. And if you have any questions, I'm sure you can email the speakers. They will be really willing to answer the questions over email. Let's thank both speakers again for the wonderful presentations. Thank you both.
Thank you.